Okay, just to start with the kind of obligatory company slide. So Oxford Nanopore, lots of you will have heard of us. Everyone we're cool, everyone wants to use our tech. The one thing I'm going to talk about today is actually on this kind of corporate slide, which is why wait real-time analysis. I won't bore you with more corporate stuff, go talk to the salespeople. So just a bit about my background. I'm kind of in customer bioinformatics now, but I was at Nanopore for 10 years. My role in the first kind of 8 years of that was on primary and secondary analysis, so base calling, variant calling, consensus calling, a bit on assembly, but not as much as Eric. Go talk to Eric, not me, about assembly. But in the last 3 years, I've kind of formed a new group around customer bioinformatics and really driving the adoption of Nextflow within the company. So my group works to standardize and automate the authorship, testing, packaging, distribution of Nextflow. Some people still use SnakeMake in the company. We don't hold that against them. My group, a lot of what we do is actually take SnakeMake and port it to Nextflow. It's really boring. I just try and stay away from doing that. It's fine to use SnakeMake if someone's already got SnakeMake. This talk's kind of an extension of something that I gave at our own conference in the summer, so if you want, you can go listen and be bored more by me as well. My group makes this desktop application, Epitome. This kind of like a desktop version of Tower. It looks very similar, you'll see as this plays through. The idea is that the company motto, sequencing anything, anywhere by anyone. The anyone and any where part of this is key. We take devices into the field. We don't have a HPC cluster nearby. We want to analyze it on a laptop. So we have a desktop application that takes you through running Nextflow workflows all the way through to here you see JBrowse being run at the end of an analysis. So as I say, this is supported on Windows, Mac, and Linux. As Evan said yesterday, science is done by scientists. So this is kind of taking what bioinformaticians are good at and giving it to the scientists without needing the bioinformaticians to get involved all of the time. So on to the meat of my talk. Why would you do analysis in real time? It's not really the normal sequencing model of collect a sample, send it off for sequencing, get the results back maybe a few weeks later, or maybe quicker if you're lucky enough to have your own sequencer. I'm going to give one example. So TB is still a big killer, but predominantly in India, Southeast Asia, some parts of Africa. And taking the experiment and the analysis close to those samples is kind of key to be able to action anything. So we want to make tools, give those to people in the field. And actually, Harsh will know this, the UK was very good at doing this during the pandemic with COVID. We're very good at collecting samples, analyzing them, sending them to Edinburgh for them to build phylogenetic trees. And so something very similar can be done with TB. Moving slightly outside of just the specifics of TB and where my talk's going to go now, we want to be able to do metagenomics in real time. So we want to be able to take a sample from the environment, detect what's there, and then provide some kind of insights. This is something that a number of years ago, our marketing people gloriously called what's in my pot. Some people changed the pot for ruder words, I'll say. There's two forms of this. You either do it on a whole sample, or you might do it with amplicons, so targeting RNA genes. And from this, you can say things about infectious disease, not just tuberculosis, but meningitis. You can do flu typing with this. Gen Z like to know what's in their microbiome, so whether they need to eat more kimchi or anything. Also, you can do, it's not just infectious disease, but also environmental studies. So one thing, again, to take an example from the UK, the water boards often get in trouble for releasing effluent into rivers. People want to monitor that and fine them and whatever. Also, in industrial food production, you might have a production line making, I don't know, pasties. You want to know if those pasties have got beef in or if someone's put pork or dog or whatever, right? So that's something you can do as well. For those not familiar with metagenomics, the basic process is you collect your sample, you you do some DNA extraction, sequencing. You take those sequence reads and then you classify them. So you say this thing is from whatever organism. For long reads, we use Kraken 2. It's kind of well benchmarked, it's fast, it's accurate. It's probably the lowest memory implementation of this sort of thing. After you've got those single read classifications, you want to try and estimate the abundance of things in your sample. So that's not a simple thing to do. There's some processing in that. And the important point here is that all of those three things to the right-hand side can be done concurrently or in real time. You don't need to wait for the sequencing to finish to perform read classification. Just to kind of go a bit deeper in this, sequencing is sampling a distribution. There's a certain number of things there in your sample, and you can take some measurements, you can observe what you've seen, and then you could react to that. So this animation is showing as a function of time, like sequencing time, we observe more and more unique species or organisms. And there's two examples here. The top one, we have a kind of flat distribution of organisms present. That's not very realistic. You don't really tend to get that. But what you see is the number of unique species that were observed kind of saturates very quickly, whereas in the bottom case, there's more kind of, some things are present to a high abundance, some things are present to a low abundance, and you kind of see this curve that kind of more slowly accumulates over time, and that's because some things are rare. You need to do more sequencing, more sampling of that distribution to detect those things. Practically, this means that if you can see that this saturation is happening, you can stop your sequencing. You don't need to sequence more and more and more stuff. You can have some confidence that you've seen everything that's present. So how do we do this? Well, we need some form of data streaming. We don't sequence, stop, do the analysis. We need to stream the data off of the device. So in the kind of Unix philosophies, here's an example. You take Minimap, you pipe it through some various processes. And Nextflow channels are very similar to this. In particular, though, there's this kind of slightly esoteric thing, watch path, which allows you to start a stream before any data is present, and then watch and monitor and see new things come. And I'll highlight this point here, which is that this functionality is one of the major reasons we chose Nextflow over other workflow managers. There's a difference here, though, is that Nextflow doesn't handle streams of data, it handles streams of files. So in the context of this metagenomics application, we have a file of reads, we put it through Kraken, we get out some classifications, we take another file, and so on and so forth. So we have from a stream of input files, we have a stream of classifications. This isn't great. Each one of these Kraken processes would take upwards of 64 gigs to hold the database of all the species. Just loading that data into memory can take 30 to 60 seconds. And the other thing we want to do is we also want to accumulate these classifications over the time. It's not the stream of classifications that we want, it's the accumulated result. So we've got two pieces of the puzzle here to solve. One is how do we keep the database in memory? The obvious way to do this is to use a kind of server-client type architecture. So we load the database onto a server. We have clients that kind of can query that server, multiple clients if we want. The way we actually implement this is to have a Nextflow process start a server and then have other Nextflow processes start clients that talk to that server. So the servers and the clients, they're not talking over Nextflow channels, they're talking over kind of network protocols. The way we actually implemented this was something called gRPC, if anyone knows what that is. The server and the client we have this as open source software. You can use this independent of our Nextflow workflows. So if anyone wants to go redo NCBI Blast with a more modern tool and wants to make Kraken, like, I don't know, NCBI Kraken, that would be the tool to do it with. The second part of this, I have to say a kind of thank you to Paolo for doing this one evening. I think it was probably a Saturday night. After explaining what it was I wanted from him, I'll get to the end, but he didn't do exactly what I wanted, but he made something that we could work with. So there's these scan processes. We're all familiar with a channel. We have an input and an output. And these recursive scan processes can take the output and feed it back into the input. So along comes another input, and we can combine that with the previous output. So in this simple case, the combination is just adding two numbers together. So 4 plus 3 is 7, 1 plus 7 is 8, 1 plus 8 is 9. That's a very simple example. In our real-world example, the input here is the classifications from each batch of reads, and the output stream is the accumulated classifications. As I said, Paolo didn't do exactly what we wanted. The implementation in Nextflow doesn't just give you the last output, it gives you the accumulating stream of previous outputs, so the entire history, which is a bit cumbersome. We only ever want the last one. And also the way tuples are handled in just the semantics and the grammar of the language don't really fit well with this scanning recursive ability. Right, so just to kind of finish and wrap this up, putting all of this together, we can run this in our application. So here we are just starting a metagenomics run, telling it to do it in real time, loading a database. This isn't with a real sequencing run. You'll see a bash window pop up here, and that's me simulating a run. So it will inject some, I think, TB reads come first. 20 seconds later, we inject flu reads into the system, and then 20 seconds later, we inject a more kind of community of things in. To start off, what happens, we see the Kraken server starts. None of the clients start until we see some reads. And then when you see some reads come into the system, multiple clients start processing those reads. And the kind of key part of this and the result that we want is to have these reports generated cumulatively over time. So first, those TB reads make it through the system. We generate a report just from those. Finally, the flu reads come through the system. We can generate a report from the TB and the flu. And then that larger community eventually comes through as well. And we kind of get our final report of all of these species. So we're progressively creating a larger and larger data set. We haven't waited for all of the sequencing to finish before we started doing this. This is obviously only a toy example. Right, so if you want to read more about this, we have a blog post here. We actually did this work quite some time ago. Just to conclude then, we have a sequencer, it works in real time, but until now we didn't really have a way of making and exploiting that. The largest nanopore sequencer can generate 64 megabases of data a second, so that's a lot of data. You can do something with that. And then Nextflow allows us to analyze this data and do something with it. So thank you to Paolo for kind of spending his Saturday evening implementing that for us. It was something that we wouldn't have been able to do this without that, at least in Nextflow. And with that, I'll say thank you.
Thank you so much for the very cool talk. I had no idea that was even possible, so that's very cool. We don't have time for questions, but if you can maybe put the link to the blog post in the Slack channel, I think that would get a lot of interest. Thank you so much.